Hi students, welcome to Vivid Botany. We are seeing here another question of the cell cycle, especially of Prophase 1 of Meiosis-I. So here 4 terms are given. I will trying to explain about the 4 terms one after the other. Let us see first the leptotin. It is the starting phase of the Meiosis-I, that is Prophase 1. So during the leptotin, chromomeres appear. There are knob-like structures present on the chromosomes. Apart from that, in the leptotin, all the chromosomes cluster and appear like a bouquet or like a knot. It's also called bouquet stage, or it is called the knot stage. So all the chromosomes cluster like a knot or bouquet. So it's called as bouquet stage, or it is called knot stage. Next one, that is zygotene. During zygotene, the homologous chromosomes will pair. What are homologous chromosomes? The chromosomes which are similar in character and structure are called as homologous chromosomes. So these homologous chromosomes pair. So this pairing is called as synapsis, SYNA, not SYNO, synapsis, and each pair is known as bivalent. Generally, the number of bivalents is equal to haploid number. For example, if you see in maize, 2n equals to 20. 2n equals to 20. So, the number of bivalents is equal to 10. The number of bivalents in maize is equal to 10 in each meiocyte. So, what are homologous chromosomes? I told you that the chromosomes which are similar in character and structure. For example, the chromosomes having one type of genes. For example, the gene that determine length of the plant are present on chromosome A, capital A. Then there will be another set of small a that is inherited from female parent. So these two chromosomes, one is capital A, one is small a, have the genes about length of the plant. Such chromosomes are called as homologous chromosomes. These homologous chromosomes will pair. Such pairing is called synapsis, and the pairs are called bivalents. And during the zygotene, synaptonemal complex is also formed here. Synaptonemal complex is formed during the zygotene. They enter into the next stage, that is Pachytene. Let us see what happens in the Pachytene. In the Pachytene, each chromosome appears to have made up of two chromatids. So like that, what happens here? One bivalent contains here 4 chromatids, because one bivalent contains actually 2 chromosomes. One bivalent contains here 2 chromosomes. So here one chromosome contains two chromatids. So total here you can find here one bivalent, two chromosomes, four chromatids. One bivalent, two chromosomes and four chromatids. Hence it is called as Pachytene tetrad. Because it is having here four chromatids, so it is called Pachytene tetrad. At the place two non-sister chromatids are fused with each other, they are called as recombinant nodules. They are called recombinant nodules. So this is also recombinant nodule. What do you mean by non-sister chromatids? The chromatids of same chromosome are called sister chromatids. The chromatids of different chromosomes are called as non-sister chromatids. So let us suppose here these two chromatids are called as sister chromatids. They are called as here sister chromatids, whereas these two chromatids are called as non-sister chromatids. They are called non-sister chromatids. So these are recombinant nodules. So what happens here? At the place of recombinant nodule, the chromatid arms are broken down and the broken parts are exchanged. This is what we call as the crossing over, or we call it as genetic recombination. So by this crossing over, variations occur in the offsprings, and this crossing over is catalyzed by the enzyme called as recombinase. It is called recombinase. So this crossing over leads to variations, and variations lead to evolution. Evolution leads to origin of species according to Charles Darwin. So Pachytene is the most elongated, most complicated and the most vital important phase of prophase 1 of Meiosis-I. Next, after Pachytene, they enter into the diplotin. During diplotin, the synaptic forces are weakened. Synaptic forces means attractive forces between the chromosomes of a bivalent. So these synaptic forces are weakened in diplotin. As a result, what happens? Synaptonymal complex is dissolved. The question can be asked here is that in which phases synaptonymal complex is dissolved? So the answer is here, that is diplotin. Another most important feature of diplotin is that during diplotin lasts for months and years in oocytes of some vertebrates. Diplotin lasts for months and years of some vertebrate. That is, after diplotin, the Meiosis-I will halted, that is paused. For so many years it is paused. Again that individual, when the organism reaches to puberty, it will restore after diplotin. So it is also very, very important. It can be asked as question. Now we will see the next one, that is diakinesis. During the diakinesis, synaptic forces are further weakened. The attractive forces are further weakened. What happens by weakening of the synaptic forces? Yes, it is utmost important. The two chromosomes are further departed. They move apart. So as they move apart, what happens here? Let us suppose I am trying to explain you with illustration. This is one chromosome having the two chromatids. This is one chromosome having two chromatids. Is another chromosome having two chromatids like this. You see here, the two chromosomes are pulled apart. When two chromosomes are pulled apart, you know that during diplotin, where they are attached, these parts are called recombinant nodules, or they are called chiasma also. So chiasma move towards terminal. When the two chromosomes are bivalent or moved apart, they can't be departed completely. Instead, they are moved very much distant to each other. As a result, the chiasma move towards terminals. This movement of chiasma towards terminals is called as what is that called as terminalization. It is written somewhere here. Yeah, this is terminalization. So synaptic force are further weakened. The two chromosomes move apart. As that chiasma move towards terminal. Chiasma move towards terminals, which is called as terminalization. See, the terminals are called telomeres. We know ends of chromosomes are called telomeres. So chiasma move towards telomeric ends is called as terminalization. And apart from that, since diakinesis is the last phase of prophase, the nuclear membrane dissolves, nucleolus disappears, ER and GC also disappear, and meiotic spindle is formed, and the total resources are set ready to form the spindle apparatus to conclude the metaphase anaphase. So, like this, prophase 1 of Meiosis-I is being framed as a question here. It is the most important concept of cell cycle. Many children do not understand it. They get confused the cell cycle. Remember, question can be framed here like bouquet stage is formed in leptotin. Synapsis takes place in zygotene. Synaptonymal complex is formed in zygotene. Bivalents are formed in zygotene. Tetrads appear very clear in Pachytene. Recombinant nodules are formed in Pachytene. Crossing over or genetic recombination takes place in Pachytene. The role of the involvement of recombinase is seen in Pachytene. And synaptonymal complex is dissolved in diplotin. Which of the stage will long lasting? That is diplotin. Terminalization takes place in diakinesis. Terminalization means movement of chiasma towards the terminals of chromosomes, towards telomeric ends of chromosomes is called as terminalization. That takes place in diakinesis. So either they may be asked to arrange them in an order like leptotin, zygotene, pachytene, diplotin, diakinesis, or else they may be asked to match the following. What takes place in leptotin? Where the recombinant nodules are formed, where non-sister chromatids attach to each other. Crossing over takes place in. Terminalization takes place in. Synaptonymal complex is dissolved in. Bouquet stage formed in. Knot stage is formed in. Because every heading, every concept is associated with some metabolic function regarding the chromosomes. So one should be very alert and very vigilant while studying the concept. I am trying to explain about these headings, these concepts. So hope this might be helpful to you in doing better for any questions that are related to the same concept. So not to forget to subscribe my channel, that is Vivid Botany. Thank you.